Okay, well, good morning, good afternoon, and good evening, folks. Before we start, we just wanted to say thank you for your participation today. We understand that institutions across the globe are only allowing 25, 50, or 100% capacity in lab, and we appreciate your time away from it. We've had almost 100 registrants for today's event, and this is going to be an incredibly informative hour. We have a lot of insights to share. We have a couple of researchers that have used our technology in their lab, and we'll dive into a little bit of their insights more towards the back 2/3 of the webinar today. My name is Christian Nelson. I'm the Global Sales Director here at Arima Genomics, and I'll be your host and MC for today's webinar, Deciphering the Mechanisms of Disease, HiChIP Application Expansion into Tissues, Transcription Factors, and Beyond. Well, we're pretty sure it's not your first Zoom webinar of 2020. We wanted to just again remind you of the structure. You'll see a couple of boxes here at the top or at the bottom of your screen, depending on how you have it set up. And what we'd love you to do is just really enter your questions into that Q&A box that you'll see on your screen at any time. If you have a question, feel free to, while it's in your mind, to type it in there. We'll try to answer as many questions as we can at the end of each of the speaker sessions, but we are a little bit tight with 3 speakers today. So we'll try to answer some of those questions in the chat and at the end with the Q&A session as well. Let's see, if we don't get to your specific question today, we'll try to do our best to copy and paste it and follow up with you afterwards from either Francesca, Lindsey, or myself or Anthony. And again, we appreciate any insights or inputs and questions that you guys would like to ask. As you can see on the screen today, we'll start with my colleague, Anthony from Arima Genomics, who's going to provide just a quick overview on the HiChIP platform. I think a lot of you are probably aware of it, have maybe used it in the past, and hopefully are thinking about using it going forward. He's going to talk about our recent expansion. Our R&D team has been up to some great work over the summer, and they're looking to just share with you today the validation that we've done around tissues and transcription factors, and share also a little bit of what's to come here in the future. We then have 2 great stories in leukemia that actually wasn't planned, but fantastic to have a deeper dive into a pretty intense disease that's affecting a lot of folks across the globe, of course. Francesca is going to share a bit of his technical experience with low input HiChIP. A question we get quite a bit is sample types, of course, but then how many cells do I need to input into your assay? And of course, we hope that a lot of folks will ask after today, tell me more about tissues. What's the amount of input tissue that I use for the assay? Then Lindsey's going to give us more of a leukemia regulation biological story at the end of the day, and then we'll have just probably only 5 or 10 minutes of questions, but we appreciate your time sticking around to the end. With that, just a quick minute overview. Wanted to introduce my colleague, Anthony Schmitt from Arima Genomics. He's our Senior VP of Science. Anthony means a lot to Arima as he was the foundational scientist behind the extremely impactful Arima HiChIP platform. He's a lead developer of Arima HiChIP and Capture HiChIP solutions, and I wish I could share a bit of what he's got up his sleeve for us for 2021, but you'll have to stay tuned to our next webinar in January or February. He's well-trained, well-published in the field, and a great person to work with. Anthony, I'll pass it to you. The floor is yours.
All right, Christian, thanks a lot. Let me share my screen here. I think.
There you go.
Perfect, thank you. Wonderful, let's see, are we working here? Yes, okay. Yeah, so thanks for the intro, Christian. I won't have to talk sort of about what I'm doing because you've already covered that, but I wanted to start with maybe a brief introduction about Arima for those who don't already know. We're a biotech company here in San Diego, California, where we really aim to accelerate the understanding of genome sequence, but also genome structure, and the role that those two play in health, disease, and species evolution. And we try to achieve this goal by facilitating access to high quality sequence and structure information via kits or services. Our first kit was launched in 2018, the Arima HiChIP kit for genome-wide HiChIP analysis. And since then, we've launched 2 different targeted genome confirmation assays, namely Arima HiChIP, which I'll be talking about at more length today, as well as Arima Capture HiChIP, which I'll briefly touch on today as well. Genome structure information can also be repurposed for an application called de novo genome assembly. We also have developed a new kit that's particularly geared towards that application, but I'm not going to discuss that much today at all. So like Christian said, I'm going to provide a super brief overview of Arima HiChIP as well as Arima Capture HiChIP, and then try to spend most of the time talking about what we've been working on with respect to Arima HiChIP. And just as a bit of motivation and introduction, wanted to tell a quick story about a recent publication from the Iafontis and the Syragos labs that came out this past year in Nature Genetics. They had performed HiC analysis on primary T cells collected from healthy subjects. What we're looking at here is a HiC interaction matrix kind of around the approximately 2 or 3 megabases around the MYC gene. Here's MYC sort of nested right in the left side of this interaction domain or TAD, and downstream are these several super enhancer regions that are not active in healthy T cells. If you do the same type of HiC analysis in primary cells isolated from T cell acute lymphoblastic leukemia patients, you can see this coalescence or the fusion really of these two once neighboring TADs into a single self-interacting domain shown here in the primary cells, as well as the leukemia cell line, and MYC expression turns on. If you kind of zoom in to this region here and you look at the underlying chromatin, if you look at, let's say, CTCF ChIP-seq data, for example, in this region, you see inward-facing CTCF motifs at the upstream and the downstream boundaries of this domain. And in the healthy T cells shown here in purple, you also see a CTCF binding site right there in the center that would correspond to the boundary of this domain that's present in the healthy T cells. However, in the leukemia primaries, as well as the cell line, that center CTCF binding site goes away, which correlates with the TAD fusion event that you see here in the HiC data. Also, if you look at K27 acetylation ChIP-seq data, there's no K27 acetylation signal at the super enhancer regions in the healthy cells, but there is in the leukemia cells. So, I think this is an important, example, not only for the discovery, but to illustrate that in the sort of linear genome paradigm, where you have, let's say, an oncogene about 2 megabases away from a regulatory element in healthy individuals, what you would see if just doing linear type of assays is upon the disease state, you would see an increase in expression and maybe transcription factor binding at the enhancer region. And then upon treatment, what they show in the study is that the expression of the MYC gene goes away, and the inhibitor of CDK7 prevents transcription factor binding, and this is sort of the end state that you would see under these different conditions. But with the 3-dimensional context, you would know that in the healthy state, the MYC gene and the enhancer elements are actually partitioned into neighboring spatial domains. Upon oncogenic transformation, you see the fusion of these two domains. Now the enhancer and the gene can come into a closer spatial proximity to regulate MYC expression. And then when you treat with a drug, the interaction between MYC and its regulatory element is abrogated. So it really helps put a bit more of the sort of how in how genes are regulated under this disease context. So briefly, Arima HiCIP, really the core chemistry that a lot of our technology is based on this core Arima HiCIP technology, and that's starting with fixed cells as shown here, two pieces of DNA that are held together in 3-dimensional space. We first digest with a cocktail of restriction enzymes, as shown here. Those ends are labeled and then ligated back together. So what used to be just DNA held together in 3D space are actually physically adjoined, and that's really how you capture that spatial proximity information via this process of digestion and religation. And then you purify away the protein, and then we have a quality control checkpoint that sort of assesses how well this sort of HiCIP process went. So I don't really have a lot of time to talk about the specifics of the different sample types and sample input requirements, but I'm happy to discuss that in the Q&A. This whole part that we refer to as HiCIP really is only 8 steps, and the whole thing happens in a single tube. It's a single tube HiCIP chemistry, which is really nice. You don't have to wash out lysis buffer or do anything that would be prone to sample loss. This happens in about 6 hours. And like I said, we have this predictive quality control checkpoint here. Now, after HiCIP is over, there's really just a few steps here where we shear the DNA into smaller pieces, and then those interactions are captured via streptavidin biotin conjugation. So these are the molecules that contain that 3D information that you want to enrich for. Those get pushed into the next step, which is adding your sequencing adapters. We have another QC checkpoint. Here, the library is amplified and then sequenced, and you're really sequencing these chimeric molecules that were generated through the proximity ligation process. We say we can go down to about 50,000 cells or less for our HiC technology. This has been published. The Akbarian lab at Mount Sinai has used our kit down to about 6,000 sorted dopaminergic nuclei from postmortem brain samples. And then we offer this as a kit or a service format. If you don't believe me, you can check out the website. We have several testimonials on our website, and we'll hear more about those who have used our kits later on in this webinar. And we have about 30 or so publications and preprints using the Arima HiCIP kit or service. Now, briefly, I just wanted to talk about Capture HiCIP and really the impetus for developing some of these new targeted genome confirmation assays like Capture HiCIP and eventually HiChIP is that not every biological question is best addressed through a genome-wide assay. You might have very specific hypotheses that need some sort of targeted solution. So first, Capture HiCIP, I just wanted to introduce this by positioning the Capture HiCIP workflow relative to the genome-wide Arima HiCIP that I talked about on the previous section. So Capture HiCIP is really built on the back of just regular Arima HiCIP. You go through the exact same HiCIP process that I showed on the previous slide of HiCIP, then library prep and PCR. And before you would traditionally sequence your HiCIP library, you simply introduce a single day workflow to enrich for any sequence element of interest, analogous to gene panel sequencing or exome sequencing. You design probes that hybridize to any element of interest. Maybe it's a subset of the promoters in the genome. Maybe it's a set of contiguous regions that may be around genes of interest. You can fish out all those molecules that correspond to those user-defined loci and eventually sequence them through this enrichment process. We've had a wonderful partnership with Agilent over the past year or so, developing an integrated workflow of Arima HiCIP plus Agilent for the capture. The way that we work with our customers on this is really you just can send our tech support team a list of genes, a list of coordinates, and we do all the probe design work on the back end for free. We can share that probe design with you through the Agilent Share Design software suite. It's a really handy web-based tool for doing these probe designs. We can work together to iterate on the probe design as necessary. And then ultimately the probes are procured from Agilent. And for all of the data that we've generated so far for Capture HiCIP, I'll just direct you to the application note that we published with Agilent earlier this year. So with that, we'll move on to some of the exciting new developments that Kristen mentioned in the beginning with respect to our Arima HiChIP platform. So what I'm going to do in this section is we've had sort of the version 1 of Arima HiChIP available now for several months. And what I'm going to do is sort of tell a story that weaves our existing data for Arima HiChIP with some of the new developments that we've been making with respect to validation on tissue samples, as well as other transcriptional proteins such as CTCF. So just by way of introduction, HiChIP was a technology not developed initially by Arima, but first published by Howard Chang's lab at Stanford. You can see over here on the left. And then shortly thereafter, a follow-on paper by my former PhD mentor, Bing Ren. This was one of kind of the final projects that I had an opportunity to work on in Bing's lab. And it's been really nice to get that hands-on experience in Bing's lab and translate that to the work we're doing here at Arima. So why use HiChIP? I'm just going to quickly go through a few of these examples. It's already been used in the literature to analyze the topology of cancer genomes and extrachromosomal circular DNA elements. Another interesting enhancers that are around GWAS loci to putative target genes that may be involved in disease pathogenesis. Here's a couple of publications of that example, as well as providing really a transcription factor centric view of genome organization during a variety of different processes like cellular reprogramming or differentiation. And from a workflow perspective, I think it's nice to introduce HiChIP by positioning it relative to ChIP-seq. That's, something that probably everybody here is more familiar with. And in ChIP-seq, you have your fixed cells shown here, your fixed chromatin. You shear that chromatin and add your antibody. You do your IP reaction. That DNA is purified, and then you make your final library and then sequence. And Arima HiChIP incorporates the same principles of ChIP-seq, except prior to the ChIP reaction, we perform our Arima HiC reaction to produce the proximally ligated chromatin that is ultimately used as a substrate into the ChIP step. So we go through the same process of digestion with a restriction enzyme cocktail, fill-in, and religation. And effectively, this proximally ligated chromatin is now the input to the chromatin shearing reaction and the IP. So at a molecular level, the molecules that are being enriched here have the DNA sequence content around your IP protein. And in the case of HiChIP, you still have that sequence content flanking the protein of interest, but you also have the three-dimensional interaction that is attached to that molecule, and therefore you get the chromatin interactions that were associated with that immunoprecipitated protein. Then you finish with your HiC style library prep, where you enrich for the ligation junctions, amplify, and then sequence. So like I said, this inherently captures that 1D protein localization information that you would get from ChIP-seq and the 3D interactions that were associated with that immunoprecipitated protein. I just wanted to point out that we have optimized shearing protocols for the Diagenode as well as the Covaris instruments. We've had this wonderful collaboration with Covaris over the past year. More details on that can be found in the poster that we did with them for AGBT this past year. And we offer this as a kit or a service format as well. Really, before I start to present any of the data, I wanted to frame some of this new data just with an idea of how much sequencing depth we're looking at for these various assays. So in our first iteration of HiChIP, which was validated against K27 acetylation and K4 trimethylation in human cells, we're looking at about 300 million or 100 million reads for K27 acetyl and K4 trimethylation respectively. That's substantially less than the 2 billion reads that are recommended by ENCODE for in situ HiC. And now, as we've developed the capabilities towards transcription factors using CTCF as a validation case, we find that our CTCF Arima HiChIP needs about 350 million reads, sometimes less. And as we pushed into the analysis of frozen tissues using mouse brain here as an example throughout this presentation, we're looking at about 220 million for the K4 trimethylation mark, as well as about 450 million for CTCF. So all the data is going to be generated using these approximate sequencing depths. So first, in our first release of HiChIP, we had this wonderful collaboration with Ming Hu's lab at the Cleveland Clinic. He developed one of the first HiChIP analysis pipelines where you feed your Arima HiChIP data into a mapping program called Feather, and then those alignments get fed into a statistical framework called MAPS, which also takes in a priori defined ChIP peaks from ChIP-seq data and identifies statistically significant chromatin loops associated with those ChIP peaks. And through our collaboration, we also built out some nice QC and other type of data visualization features, like we'll take all of your HiChIP data and we'll create a meta plot around the ChIP peaks. We will output all of the loop calls as these arc plots, so you can just conveniently drag and drop them into the WashU epigenome browser to view your HiChIP data alongside your other omics data sets in a single browser, as well as a really robust QC table to dive into the HiC, the ChIP, and the overall HiChIP features in your sequencing data. And what we're releasing today is now the capability of being able to call ChIP peaks with the HiChIP data. And that really enables the technology to be sort of a truly multi-omic assay where you can identify protein localization sites and their associated chromatin interactions in a single assay. And we've been using MAX2 for that. We've also looked at other different tools that have also tried to call ChIP peaks from HiChIP data. And really the reason why we can call these chip peaks from the HiChIP data is because the chip enrichment signal in the HiChIP data is quite strong. So if you do a metaplot analysis of our K27 acetylation or our K4 trimethylation HiChIP data from our first release of HiChIP, you see very nice enrichment centered on all the chip peaks. And with the new CTCF capabilities, we see the same thing, really strong enrichment centered on all the chip peaks. And as we've also validated in the mouse brain tissue for both K4 trimethylation and CTCF, again, very strong enrichment centered on these chip peaks. So the chip enrichment signal, which is really important for this sort of multi-omic aspect of the data, is quite strong across cell lines and tissues and different protein factors as well. And a way to sort of quantify the performance of the chip peak calling is if you call chip peaks and you ask what fraction of ChIP-seq defined peaks can be called using the underlying Arima HiChIP data. Well, it turns out that's about 75 to 80% for K4 trimethylation and about 70 to 80% also for CTCF. So about 70 to 80% sensitivity. And again, about 85% positive predictive value for the K4 trimethylation mark and about 70 to 75% positive predictive value for CTCF. So in sum, I think we've been really pleased with not only the chip enrichment, but also the optimized bioinformatics capabilities for sensitive chip peak calling. In the next few slides, I'm going to show examples, basically screenshots of what some of this data looks like for the various different sample types as well as protein factors. So this is the MYC locus in human lymphoblast. Here's MYC here. I wanted to direct your attention down to the bottom two tracks where we have our K27 acetylation ChIP-seq data. As you would probably predict from the past few slides, if you plot the HiChIP data as sort of this 1D signal track like you would for ChIP-seq, the peak positions and the relative heights very much recapitulate what you see via ChIP-seq. So you see this equivalent enrichment at the MYC locus. And then of course, what you're not getting from traditional sort of 1D assays like ChIP-seq is all the interconnectivity information or all the statistically significant chromatin interactions that are anchored at those K27 acetylation sites, which is what you can get from HiChIP. And you can probably appreciate here that the reproducibility between our replicates looks pretty good. When you quantify that at a genome-wide scale, what we're finding is about 84% of the loops that are observed in Rep1 are also observed in Rep2 and vice versa. And if you do this analysis for our K4 trimethylation, it's about 74 to 87% reproducible using the same type of metrics. And now extending into CTCF, this is the same type of a snapshot, but now I'm looking at the HOX-B locus, again in human lymphoblast. Same sort of thing here. I want to direct your attention down to the bottom. This is the CTCF ChIP-seq data downloaded from ENCODE, as well as lots of other omics data sets across this locus. When you plot the CTCF Arima HiChIP data, both replicates, again, you can see the nice correspondence between the ChIP-seq data as well as the HiChIP data in terms of the signal enrichment. And again, here's the interconnectivity now between CTCF peaks that you find from the MAPS pipeline in human lymphoblast cells. You can go through the same type of reproducibility analysis that I did on the past. And here, the reproducibility between these two replicates for the loop calls is about 77 to 80% reproducible. And I thought what would be interesting here is also to ask, lots of the loops that you identify using Hi-C are also anchored by CTCF at both sort of the loop anchors. So you can ask what fraction of those Hi-C based loops can be identified using CTCF based Arima HiChIP. And this is an important type of analysis because traditional Hi-C analyses, like I said, are 2 billion or so reads according to the ENCODE recommendations. And here, the CTCF HiChIP is about 350 million reads. So, what does that look like? Well, it turns out that if you download the loops from the Rao et al. Study from the Aiden lab at Baylor, it turns out that about 70 to 75% of those loops can be picked up using CTCF Arima HiChIP. Sort of really enables you to drive down the cost of identifying these CTCF mediated loops in the genome. And the last shot here I'm going to show is the ADGRB1 locus. This is a gene that's associated with angiogenesis inhibition, specifically in the brain. You can see that right here. There wasn't pre-existing chip tracks for this whole brain tissue that we validated in, but you can appreciate just by looking at the 1D signal tracks of the K4 trimethylation HiChIP data or the CTCF HiChIP data that the replicates are reproducible, and you see this really nice enrichment and low background. And then when you layer on some of the loops that are called, the blue corresponds to the K4 trimethylation based loops. If you anchor on this point at the ADGRB1 promoter, you can see lots of significant interactions sort of scanning upstream until it hits these other CTCF sites shown here. Again, quite nice reproducibility between replicates for the K4 trimethylation HiChIP as well as the CTCF based HiChIP. And when you quantify that at a global scale, again, we're sort of in the range of 73 to 86% reproducible loops for the K4 trimethylation, and a little south of 70% up to 85% for the CTCF reproducibility. So that wraps up sort of the brief update on some of the validation data. Our HiChIP has been used by several in the community. You can go to our website for some of those testimonials to the data quality, the reproducibility, the utility, and the application in various different disease or basic research contexts. So in sum, HiChIP is an NGS-based assay from multi-omic analysis of both chromatin protein binding as well as their associated interactions. I've shown that in most cases, the reproducibility between those loops that we identify with active chromatin or CTCF using cells or tissues is approximately 80%, plus or minus maybe 5 to 10%. We've really tried to be thoughtful about creating open source, easy to use bioinformatics tools, now with the new capabilities of calling chip peaks from the HiChIP data, as well as some other usability features like command line tools. I've spoken to the extensive validation across protein factors and cell types from our customers, and we'll hear more about that in just a moment, and the dedicated end-to-end tech support for those who have worked with Arima. You know this, with expertise really in the HiChIP assay as well as the data analysis phase of the projects. I want to thank the team at Arima, our scientific advisors, and the generous funding support we've received from the NIH. And I'll pass it back to Christian if you want to speak to this final slide. Thanks.
Sure. Thanks, Anthony, for the great content. Just wanted to share, we launched a couple of promotions, end of the year promotions yesterday, actually. And so just wanted to share this with all of you in attendance today. Obviously, we have folks across the globe, and so can't provide like specific details here, but we do have two places at the top. sales@arimagenomics.com for an email, or just our website. There's a box on the top right of our website where you can request a quote. If you haven't used Arima products yet, we have trial offers available through the 18th of this month. Just send us a note and put new to you in the comment section. If you have actually used our kits before, we appreciate you. We also wanted to provide you guys just a stock up discount for the end of the year, which also expires on the 18th of December. So please either email us, email your sales representative if you're aware of Brenda or Carlos out there. Feel free to email myself or go on the website and just request more information there. We appreciate it. Let's move on here to our next speaker. We're just a little bit behind, so I just wanted to dive straight in with Dr. Francesco Boccalate. Francesco is a postdoctoral fellow in the Iofantis Lab at the NYU School of Medicine and Perlmutter Cancer Center. Francesco works on epigenetic regulation of gene expression in tumors with a particular focus on T-cell leukemia. Francesco, the floor is yours. Thank you and welcome.
All right, thank you very much. Let me share my presentation. Okay. Okay, here we are. So good morning, everyone, or good afternoon, depending where you are. I'm going to show you some validation that we have done on low input HiChIP on leukemic cells, which is what we have been working on in the past few months. I just wanted to give you a brief overview. Like our lab is, I'm part of a bigger lab, which is the iFantis lab, where we work on different aspects. We have a lot of interest in both like microenvironment, RNA binding protein, drug resistance, but also recently, as Anthony has actually showed you already, we had published papers in 3D chromatin conformation in cancer. So I won't go too much into detail of this, but it's just to tell you that we are actually investing more in this technology for future experiments. And so this is, working with Arima has been very useful for us. What we're aiming at now is basically to identify how 3D chromatin structure can reveal differences in health and disease. So now we are using HiChIP in comparing leukemia samples and healthy donors, because we want to see whether we are able to capture loops that are either stable between samples of leukemia or healthy, or that may be gained or lost in one of the two conditions. And of course, this has as a final outcome is we want to evaluate specific regions of interest that we know that are involved in disease, but also discover new regions that may be important for disease evaluation, and maybe also discovering new therapeutic targets that we can actually validate by either drugs or by CRISPR technologies. I just wanted to give you a very brief introduction on the methods that are available to visualize 3D chromatin structures. So we started actually historically with more low throughput technologies, where you fragment your DNA and you capture the loops and you can amplify them by qPCR. This is the 3C technology, or you can sequence them by 4C. But as you know, like you can go deeper than that, and you can either capture these loops with biotin beads or with specific pulling down, for example, chromatin markers like K27 acetylation or methylation. And this is actually HiChIP, which I'm going to talk more about. And in terms of what you can see with these technologies, as most of you are aware of, like they give you different resolution, they give you different outputs. So with 4C, you basically study the interaction of just one locus to many neighboring loci. With HiC, you have a broader vision because you can capture all to all. Whereas for HiChIP, depending on the marker that you're using, you can capture many to many, as is in the case, for example, of K27 acetylation. That is what we have used. This is something that we have done. We wanted to compare because, as you can imagine, like both HiChIP and HiC can be used to identify enhancer promoter loops, but which one is better? So to do that, we worked on some samples from a collaboration. These are ESCs, and we plotted the interactivity quartiles versus the gene expression, and we saw that by promoter interactivity, it's quite clear that although both perform pretty well, HiChIP gives you a better resolution in terms of for higher quartiles, so where the promoter interactivity is higher, it correlates much better with gene expression. So this is why we decided to go further and invest more in the HiChIP technology for this specific question. And so to do this, we took a TALL cell line, and we wanted to compare what happens if we use the recommended dose or a lower initial amount of cells. This is simply, that's not a trivial question, because when we work with clinical samples, most of the time we don't have 3 million cells to cross-link. So we wanted to basically scale it down and see whether we were getting good results anyhow. So we directly processed, like in parallel, these two samples following the Arima HiChIP indication. We had to optimize sonication, and this, like Anthony helped us a lot with this. And then we immunoprecipitated with the recommended amount of antibody, and we checked chip efficiency and QC. As you can see, like the two samples that are directly comparing is this leukemia with 3 million and leukemia with 1 million. As you can see, the yield is actually kind of similar in terms of chip QC. As you see, the chip efficiency is pretty high comparing to what Anthony was telling us, but also it's because here we were using an antibody that was not the one was recommended by the kit. This was an AppCam antibody that is probably a little more dirty. This is, I also want to show you that when we did other samples with the active motif, HA27 K27 acetylation antibody, which is the one that Arima recommends, actually the chip efficiency is below 1, which is what Arima is expecting from these samples. But anyhow, like all the samples were good. They passed the quality control, so we were good to go with them. So I'm going to show you directly the comparison of the 3 million versus 1 million, as I said before. This is what we got in terms of sequencing reads. As you can see, both the 3 million and 1 million, we got around 100 million reads. And of the useful reads for analysis, the accepted identifier reads, these green ones, they were about like little less than 40 million reads, and there's definitely no difference in the 3 million or 1 million, neither if you look at count or percentage. However, if then you process the samples and you calculate the loops with K27 acetylation HiChIP, you actually see that there is a big difference. So if you calculate loops with a 10 kb window or a 5 kb window, it's not a big difference, but you can definitely see that you have almost double the number of loops identified with 3 million cells than with 1 million cells. We also tried to merge them to have like a more comprehensive picture, but let's focus on the two separate ones to calculate the differences. Yeah, so as I said, this is kind of was a concern because we say, okay, we're picking up basically double the amount of loops. So how are these loops distributed? If we do a Venn diagram to compare what the loops that we get with 3 million and with 1 million, the comforting fact is that the majority of the loops that we get in the 1 million are contained in the 3 million. This means that you're not losing much by using 1 million, but of course, if you use 3 million cells, you pick up more loops than what you're able to do with just 1 million cells. So, but the other question that was interesting to us was, is there a bias in these loops? Like, why are these like so different? So we tried, well, first of all, let me show you that on a locus of interest that we had on HD19, this was not a big deal because we were able to pick up by setting the viewpoint on the gene that we wanted to see. We were able to see the peak both in 3 million and 1 million, although it was a little taller in the 3 million, but still the loop was picked up. But more on a general point of view, so looking at all the loops of the sample, we tried to plot the common loops based on loop strength to see what was the difference between 1 million and 3 million. And the first analysis that we tried was a quartile distribution. And as you can see from here, from a theoretical distribution, you can definitely see that the comforting part is that for looking at the CPM comparison, most of the samples in the first 3 quartiles, they are actually on the diagonal. So they're pretty much in common. They're distributed equally. A more refined analysis of this one is by using hex bins. So we tried an hex bin distribution, and this is basically like each hex bin is a group of, each dot is a hex bin, so a group of loops. And as you can see, it's giving you a similar result because we have the vast majority of the stronger loops, with the one with the stronger counts, above 100, they are actually on the diagonal, whereas the weaker ones, they are more present in the 3 million reads. Well, as you see, like the R is actually pretty good. So this is telling you that the bias is not big. However, you should keep in mind that if you use only 3 million cells, you are capturing also weaker loops, whereas the stronger loops are captured by both. So if you're interested also in loops that are weakly, not strongly represented, you should go for the 3 million, of course, given that you have enough cells to do that. And also another thing that we tried was we wanted to normalize this with previous samples that we had in the lab that were done with HiChIP with the same antibodies. However, the number of loops across these samples, just look at the red numbers. So for the control CD34, we have 500,000 loops, whereas with controls here we have a similar one. But for example, in the TALL, these were way less. So what happens if you want to try to compare samples that have a high variability in the number of loops? We're still working on that bioinformatically. One idea that we came out with was using fractions. So basically dividing them by loop fractions. So let's say the strongest loops were divided into bins, and then we give them a threshold when we reach like more, this is an arbitrary threshold, but we try to consider the same threshold for all the samples. And this allows you to normalize the samples a little better, because although here there's like a 5-fold difference, if you use this method, you're actually able to normalize a little better the samples by dividing them into bins and then setting a threshold here. This is still ongoing. It's just to show you like what we're trying to do. So my conclusions from this analysis was that we tested that HiChIP is more efficient than HiC to detect enhanced or promoter loops, and that HiChIP is efficient even if you have low input material. However, if you have less material than recommended, you can have a slight bias towards stronger loops. We have also tried in the lab using less material than 1 million cells, and this was kind of forced because one of my colleagues was trying to run the analysis on a precious clinical sample that we were not able to get 1 million samples from. So, but for this one, we haven't been able to sequence it yet, so I cannot tell you whether the analysis worked out pretty well. I can just tell you that the HiChIP per se, in terms of the Arima quality controls, was okay, but we don't have the loop analysis yet for that one. So as for future directions, we want to develop new pipelines to normalize samples, as I showed you briefly before. They're still ongoing, so if anyone has recommendation or suggestion, is more than welcome. And also we are thinking of trying also new protocols for experimental normalization if we have samples that are coming like from different sources. One of these ideas may be this new publication is this HiChIP, where you basically use a spike from a different organism and you merge it with your sample, and this is used as a normalization of the material that you input into your samples. We haven't tried this yet. If anyone has experience, feel free to reach out. We're more than welcome to discuss that. And I just wanted to acknowledge briefly, this is a part of a much bigger project that involves so many people and collaborators. But I also wanted in particular to thank Arima Genomics, because they have been very helpful to set up our experiments and to troubleshoot some things, especially at the beginning. And also thank all of you for your attention.
Great, thanks you, Francesco. We appreciate your time and insights, and understanding you have to scoot across town here shortly. I'll just ask everyone just a little reminder, I put it in the chat as well. to add any questions you have in the Q&A box. And if they're for Francesco and he's not available to stick around, then we'll definitely get those answered and back to you shortly after the webinar. Thank you, Francesco. Next up, just with the eye on the clock, we're a little bit behind schedule. Hopefully you can stick around until maybe 5 or 10 past the hour to hear from Lindsey. Next up is Lindsey Montefiori. Lindsey received her PhD from the University of Chicago in the Nobrega lab of the Department of Human Genetics, where she used promoter capture HiC to map cardiovascular disease GWAS loci to target genes. She is currently a postdoctoral fellow at the Charles Mulligan Lab at the St. Jude Children's Research Hospital, Department of Pathology. Lindsey is focused on modeling genomic alterations in lineage ambiguous leukemia. Lindsey, thank you and welcome.
Thanks, Christian, and thanks all so far for this great webinar and the opportunity to speak and share our research with you and how some interesting insights we've been able to make with the HiChIP kit. So I am a postdoc in Charles Mulligan's lab at St. Jude, as Christian mentioned, and our lab studies acute leukemia. Which is, as you all probably know, characterized by the uncontrolled proliferation of hematopoietic cells. And there are many different subclasses of acute leukemia. And to try to summarize a main issue with diagnosis and classification, I'll just point out that leukemias are classified and diagnosed based on their phenotype and their morphology and how similar that is to normal hematopoietic lineages as shown here. But there's this relatively rare but very high risk group of leukemias that are called lineage ambiguous leukemias. And these are thought to arise from the most immature hematopoietic stem and progenitor cells. And they're called lineage ambiguous because they express markers associated with both the myeloid and lymphoid lineage. And this is problematic in terms of figuring out which treatment to give these patients. And of course, as most cancers that have stem cell characteristics, they're quite resistant to chemotherapy. Recently, our group and others have turned to transcriptional profiling as a way to dig deeper beyond just cell surface markers and morphology to investigate biological heterogeneity within leukemias and other cancers. And last year, we published a paper that focused on B-ALL. So this is showing T-SNE projection analysis of nearly 2,000 primary B-ALL leukemia cases clustered by gene expression. And they're colored based on their genomic alterations. And so what you can immediately see is that there are very distinct gene expression clusters that emerge, and these are driven by genomic alteration. And in this paper, there were 23 subtypes described, and this has important implications for a lot of things. So, these different patients with different alterations have different risk assessment and should be stratified differently for their treatment. They have different responses to therapy. And then in terms of the biology of the leukemia, obviously different genes and pathways are affected, and this can lead to subtype specific differences in vulnerabilities or dependencies. And this is important for future sort of personalized medicine approaches in leukemia. So the work that I'll show today was motivated by taking an approach like this, transcriptional profiling, to identify biologically distinct groups within this high risk and lineage ambiguous group of leukemias. And so I'll just jump right into the results. This is a T-SNE plot showing the results from 1,162 primary samples spanning the the lineage ambiguous leukemia spectrum, as well as canonical T, ALL, and AML. And so you can see there are distinct clusters that emerge, and these are characterized by genomic alterations. But the main player in the story today is this group here, which had not been previously observed. This is a very distinct gene expression profile, and all of these samples are characterized by monoallelic expression of a gene called BCL11B. These samples also all harbor structural variants that were largely identified from whole genome sequencing data involving the BCL11B locus. And now I'm coloring the samples based on their original diagnosis. And it's important to note that this group is comprised of multiple diagnostic entities. And so this is, has implications just for how leukemias are classified and treated, but is kind of beyond the scope of the talk today. So for the rest of the time, I'm just going to focus on the interesting genomic events that characterize this gene expression group. So as I mentioned, all of these samples harbor structural variants or rearrangements involving the BCL11B locus. So here I'm showing you this locus. It's positioned towards the telomeric end of chromosome 14, and it's neighboring a very large gene desert. The BCL11B gene itself is about 100 kilobases long, and each breakpoint from these patients is shown grouped by the rearrangement partner. So the most common group is shown at the top, which is rearrangements to the ARID1B locus on chromosome 6. And then I'll just highlight that the only group of rearrangement that does not involve another chromosome is a tandem amplification event that occurs 700 kilobases downstream of BCL11B that I'll come back to a little bit towards the end. And if we zoom in on this locus a little bit more and sort of compile all of the breakpoints into one track, you can see that all of these breakpoints spare the coding region of BCL11B, except for this SEB2 BCL11B fusion, which occurs in intron 1. And so this was kind of our first indication that these rearrangements are probably moving BCL11B somewhere else in the genome, so not mutating the gene or altering its function, but targeting its regulation. So BCL11B is a transcription factor, a zinc finger transcription factor that plays a very critical role in normal T cell differentiation. And in this plot, it's showing the expression level of BCL11B throughout hematopoiesis. And you can see that it's highly expressed pretty specifically in T cells. However, the hypothesized cell of origin of lineage ambiguous leukemia is a hematopoietic stem or progenitor cell. And you can see here that BCL11B is not expressed in these cells. So that together with the monoallelic expression and led us to hypothesize that these structural variants are leading to ectopic BCL11B activation in a hematopoietic stem or progenitor cell through enhancer hijacking. So to initially investigate this, we just looked at publicly available K27 acetylation SHIP-seq data in hematopoietic stem and progenitor cells, which can be summarized by cells that express the CD34 cell surface marker. And so this is showing the chromosome 6 locus, which is one of the most common breakpoint partners. And highlighted here are the positions of breakpoints, and they all cluster around this very strong enhancer, which is also classified as a super enhancer, shown by the red bar. And sure enough, we found that every other rearrangement locus observed in this group contains at least one super enhancer present in hematopoietic stem and progenitor cells. And so based on this information, we sought direct evidence that these structural variants lead to chromatin interactions between these CD34 super enhancers and BCL11B. And so of course, for this, we turn to the HiChIP method, which has been introduced, and we used H3K27 acetylation as our target. So I'll just show the samples that we used were sort of, from different, so healthy normal cord blood cells where BCL11B is repressed, and this represents sort of a fresh primary sample. So these cells are happily growing in culture at the time of cross-linking. We also generated data from 5 primary cryopreserved leukemia samples that were taken at the time of diagnosis. And so these are frozen cells. Sometimes they had been frozen for over 10 years. The viability can be poor. And so we were curious to see how the HiChIP kit worked for these different situations. And then finally, we also generated data in 2 T-ALL cell lines. So a spectrum of sample preparations, and I'll just say I won't show the results, but all of the data look really good. And so I was very, we were very happy at the performance of the HiChIP kit, especially how well it performed in these primary samples, which are pretty precious to work with. And we sequenced our libraries to an average depth of 370 million reads, and the data were processed with the Arima Feather MAPS pipeline. So this is results from healthy, normal hematopoietic stem and progenitor cells from cord blood. And so this is leading into showing the data for a patient with the chromosome 6 rearrangement. rearrangement. So I'll first just show what the data look like in normal cells. On the left is the chromosome 14 locus harboring the BCL11B gene. On the right is the chromosome 6 locus harboring ARID1B, and this is that strong super enhancer. And in normal CD34 cells, you can see that there's evidence of this enhancer looping to ARID1B promoter. But on the left in the BCL11B locus, where this gene is repressed, there are no chromatin interactions because this gene is not active at this time. However, in the primary patient sample that harbors this rearrangement, you can see that there, first of all, if you look at the bottom, just comparing the one-dimensional enrichment track or signal track, you can see that there's now very strong H3K27 acetylation signal in the patient sample as compared to normal CD34 cells. And this is also shown that this is probably due to enhancer hijacking, which is what's shown by these black arrows. This is evidence that these strong enhancer peaks on chromosome 6 are directly interacting with the BCL11B locus on chromosome 14. And I just want to point out a couple of interesting observations, beyond this enhancer hijacking evidence. If you just look at the BCL11B gene itself, these are cis interactions over this gene, which again is it's about 100 kilobases long. And there's this sort of mini TAD that has formed over this gene. And we thought this was really interesting because when you compare this region to, sorry, my animations have messed up. So if you compare this region to and JERCAT T-ALL cell line, which also expresses high levels of BCL11B, but this cell line is derived from T cells, and BCL11B is normally expressed in those cells. You see that you don't have this K27AC signal throughout the entire gene. And so we are speculating that the mode of activation of a gene that's normally very tightly repressed by this very strong enhancer coming in sort of out of nowhere might lead to possibly a different mode of activation, and that's reflected in this very broad K27AC domain that we see. So more of just an interesting observation from the HiChIP data. So, the way that I'm showing the data here is between two different chromosomes. And so these are, because these data were mapped to the reference genome, these are interchromosomal read pairs. So these are reads where one end maps to chromosome 14 and one end maps to chromosome 6. But of course, in the patient sample, the translocation generates, that these reads are actually in cis if we were able to map to a genome that reflected the patient's genome. And so we were interested to do that because we wanted to, get an accurate view of genome conformation of the BCL11B locus with its rearranged configuration, as well as to call loops, because most loop calling algorithms they throw away interchromosomal reads. And so you're not able to call loops between two different chromosomes. So I'll spend just a few minutes discussing our approach to generate patient-specific maps. So of course, in the reference genome, there's 2 copies of wild type chromosome 6, 2 copies of wild type chromosome 14. But if we zoom in on the alterations in this one patient, what I'm showing you here is the position of the breakpoint for the patient I just showed the HiChIP data for. It occurs 30 kb downstream of BCL11B. You can see the very strong K27AC signal over this gene compared to normal CD34 cells. And the other breakpoint occurs right at the end of this strong enhancer at the ARID1B locus. And so we know from the whole genome sequencing reads that the orientation of the rearrangement places the centromeric end of chromosome 6 at the telomeric end of chromosome 14. And so the way that this configuration actually should look like is something like this, where the ARID1B enhancer is immediately downstream of the BCL11B gene. And this just simply isn't reflected in this sort of traditional way of showing these data. So to generate patient-specific genomes, we first generated new, a hybrid FASTA file for each of the rearranged chromosomes. So that's just the file containing the sequence, the actual sequence of the chromosome. And then add that to the remaining chromosome FASTA files. And then importantly, we had to remove the wild type chromosome 6 and chromosome 14, and that's to prevent reads mapping to 3 locations. And also now you would introduce sort of a competition for mapping between these the wild type chromosome 14 or the derived chromosome 14. And so I think that could be an interesting discussion to have in the future about how to more accurately maintain mapping when you have a hybrid chromosome present in the genome file. Then, and I'd just like to point out that a lot of this was guided by instructions provided in the MAPS and Arima bioinformatics packages. So I had to generate a new genomic features file for this rearranged genome. So just recalculate mappability using a program called GEM and convert to BigWig to be compatible to run with the genomic features generator scripts, which were provided in the Arima MAPS pipeline. And then also, because HiChIP requires, or analysis requires a ChIP-seq-peak file, I just converted the ChIP-seq-peak coordinates to reflect the derived positions, and then rerun the pipeline using the new genome file. So it's pretty straightforward. Just takes a few days to run, and of course you have to generate all of that by hand. And so I'll just use this as an opportunity to say that I think with a lot of interest in analyzing genome structure in cancer genomes, this is something that would be really useful to have a program that would streamline this entire process and take some of the user interface out of it, or specifically having to generate these files by hand. So this is what it looks like after mapping to the faithful genome of this patient sample. So now you can see that the ARID1B enhancer is downstream of BCL11B, and there's really strong, really nice visualization to see that there's looping between these chromosome 6 derived enhancers and the entire BCL11B gene. And so I'll just quickly say that we did this for a couple of other patient samples, and I'll show just one. So this is an interesting sample that contains a rearrangement to a locus that has been mentioned earlier today. So the MYC locus on chromosome 8, which I'm showing down here, contains a strong super enhancer that's over 2 megabases away, called the blood enhancer cluster, or BENC. And quite a substantial proportion of patients in this expression group contain breakpoints near this enhancer. And so this results in this enhancer now looping and regulating BCL11B. And so after mapping to the patient-specific genome, we can see, again, very clearly evidence of interactions between these enhancer peaks actually spreading down into the neighboring region and then interacting specifically with BCL11B. And we note that there's kind of this dearth of interactions in between the BCL11B gene and this next gene, set D3. We've seen this in all of the patient samples, and we're not really sure why that is, but one speculation is that there's a boundary element here that prevents this continued spreading of active chromatin marks. But then these interactions pick right back up and are able to activate BCL11B. So I'll just quickly mention this final genomic event for the interest of time. So this is about 20% of these patients harbored a tandem amplification of a 2.5 kb non-coding region that's located 700 kilobases downstream of BCL11B. We identified this from whole genome sequencing. So as you can see, these are patient data from samples harboring this amplification, and the whole genome sequencing track shows this large peak here. And in contrast, a sample that doesn't contain this amplification is relatively flat. So HiChIP data has shown that there's a very quite a massive signal of K27AC on this region in 2 cases harboring this amplification. In contrast, 3 additional leukemia samples that do not have the amplification do not have any K27AC here. And in the CD34 cells, as well as both of the T-ALL cell lines, there's no K27 acetylation. And so this indicates to us, we don't know what this region does normally, but clearly after tandem amplification, it becomes, a very strong enhancer. And so the actual HiChIP data shows that it is specifically interacting with the BCL11B gene that's shown as highlighted here. And I should say we've termed this the BCL11B enhancer tandem amplification. So that's why this is called beta. And significantly called loops from the MAPS pipeline calls interactions that span the entire BCL11B locus. And just contrasting to two other samples on the right are data from the T-ALL cell line, where you can see that there is no evidence of interaction between BCL11B and beta. In contrast, there's a very distal enhancer that's active in normal T cells called ThymoD. And this is the enhancer that's driving BCL11B expression in this cell line. But this enhancer is not active in primary patient samples. So in conclusion, I showed you that transcriptional profiling has identified a new subtype of lineage ambiguous leukemia, which is defined by BCL11B deregulation. H3K27 acetylation HiChIP data demonstrated enhancer hijacking of CD34 hematopoietic stem and progenitor cell superenhancers in 5 primary leukemia patient samples. I walked you through sort of the pipeline that we've taken to map data to genomes that reflect the structural rearrangements that we've detected from whole genome sequencing data. And I think this will be a very useful approach just more generally in cancer genomes. And then this final topic of this tandem amplification that we've termed beta that may represent a new mode of superenhancer generation in leukemia. And I would just like to thank everyone who's involved. This is quite a large project and there are a lot of people involved. But just to mention my PI, Charles Mulligan, as well as former postdoc Xiaowei Gu, who's done a lot of the gene expression work. So sorry for going over, but if there's questions or discussion, I'm happy to stick around.
No worries, Lindsey. Thank you for everyone for sticking around for an extra 5 or 10 minutes. Actually, I have a couple of questions here that are more technical in nature to start for yourself, Lindsey. How did you cross-link the cryopreserved cells?
Sorry, I cross-link?
Yep. How did you cross-link the cryopreserved cells?
Oh, right. So, we thought a lot about this, and what I ended up doing is exactly what is recommended in the Arima HiChIP protocol. So just thaw the vial very quickly and dump it into 4 mils of PBS, and then quickly get a count. But regardless, I just went forward with it. So the cell count ranged from 1.5 million to 8 million cells. The viability ranged from maybe 20% to 60%, and then proceed with cross-linking 1%. So no spinning.
Okay. And maybe the follow-up question is kind of along those lines. Do you have to remove dead cells prior to the HiChIP assay?
Yeah, we didn't. We definitely thought about it, but like I said, I actually think some of the cleanest data was from the primary leukemia samples, and that chromosome 8 rearrangement sample was a very poor viability sample, but the data looked, pretty sharp to us. So I don't think that it affected the quality that much.
Okay. Good stuff. Last one here, I think, is would a haplotype resolved analysis help your analysis, given that the structural variants involve just one allele?
Yes, I think that, and that's something that I've also talked to the technical support team about. I think that would help a lot, depending on how dense haplotype information would be between the alleles and surrounding the breakpoint. That would solve the issue of being able to keep the wild type copies of in the cases of reciprocal translocations, definitely.
Okay, great. Appreciate the insights. Just checking the Q&A box again. It looks like these questions might be more for Anthony. Just give it another second here. Any other questions for Lindsey or Francesco? Okay, I'll move on. Thank you, Lindsey. Appreciate it. If you could stick around, that'd be fantastic. We have a couple of questions here for Anthony. I'll start at the top. For Capture Hi-C, instead of designing new probes, does Arima have a set of predefined probes for promoters of how many genes? Promoters of genes, sorry. Is that clear, Anthony?
Yeah. We have pre-designed panels that target I think it's about 26,000 or so protein coding and non-coding gene promoters in both human and mouse. I think those can be more or less obtained off the shelf, so to speak. And then of course we can also do custom designs for a subset of gene promoters or other defined regions, depending on what you're interested in.
Okay, great. Question 2, looks like there's 4 now. Have you tried SMC1A with your HiChIP kit? And also, which CTCF antibody works best with your kit?
Yeah, there was another question I think I answered regarding cohesin and cohesin subunits. I think we tried RAD21 and SMC3. Not so sure we've tried SMC1A yet, but we have seen the best results with RAD21. We spent most of the product development time focused on CTCF, but we have produced a successful library with RAD21, and then not quite as good with SMC3. Oh, and then for the CTCF antibody, I believe we're using the recombinant antibody from Active Motif, but I can double check and follow up afterwards.
Okay, great. Yeah, I'll record the question here. Let's see, question 3 of 4. Since Arima HiChIP pipeline maps requires ChIP-seq information, do you recommend using external ChIP-seq data or self-called ChIP peaks from HiChIP pipeline for calling significant interactions?
Yeah, I mean, I think the idea was that with some of the informatics developments that we've done calling ChIP peaks from the HiChIP data, if you have strongly enriched, pretty clean HiChIP data, the idea would be that would be sufficient. I think if you already have ChIP-seq data, and let's say your HiChIP signal was not quite as clean or as enriched, then I think using the ChIP-seq data would still be the way to go. But if you have evidence of a high-quality HiChIP data set, then I think using the HiChIP data is fine.
Okay, great. I think the last question. This might be for Lindsey. It might be for Anthony. I'm not positive. Have you looked for CTCF between TEDs? I'm not sure who that was directed to, so sorry for the kind of open question between the 4 questions here.
It was probably me, I guess, because I showed the CTCF HiChIP data. I can say no. We haven't specifically looked at CTCF mediated loops between TADs so far. If there's like a specific biological insight that someone's trying to gain from that, Randall, maybe we can follow up afterwards.
Sure, sounds good. Any final questions? I know we're 15 minutes over, so thanks for sticking around. We appreciate it. And if not, just wanted to say thank you again to Lindsey and Francesca. We appreciate your time. Of course, the opportunity to work with you further. Looking forward to doing that over the course of a little less of a crazy year next year, hopefully in 2021. And Anthony, it's always great to get a peek behind the covers at Arima Genomics. We appreciate everyone's time, and thanks for sticking around for the extra 10 or 15 minutes. You guys have a nice day. Cheers.